undergo various checkpoints that allow them to move to the next step. So for example, the double positive cells undergo something here which is called positive selection. Positive selection means that the double positive cells are checked to see whether they can bind MHC and antigen, meaning do they bind antigen when it's bound to MHC? So they have to be able to recognize MHC. If the double positive cells are able to recognize MHC, they can progress to become single positive cells. If they can't, they undergo apoptosis and they die. So that's called positive selection. It selects cells that are MHC restricted, and that's how we get MHC restriction within our T cells. The single positive cells eventually undergo a checkpoint, and that is called negative selection. And in negative selection, what happens is the question is asked, do the single positive cells bind self-antigen? If they avidly bind self-antigen, then they undergo apoptosis, and if they don't, then they're allowed to progress and exit from the thymus. Now, if you think about this, negative selection is very important because by asking the question, do you bind self-antigen, and then killing off those cells, we're actually removing self-reactive lymphocytes. Remember, the T cell receptor was generated randomly. It could be specific for self-antigen. If it binds self-antigen within the thymus, then those cells will die via negative selection, and we won't have to send them into the periphery so